Assalamu alaikum and very good morning to all of you dear students. I hope you all are fine at your home and doing studies also well. Okay, we are learning class 9 science biology. In that we are doing the practicals, and in previous video I gave you the mind map of chapter number 5, the fundamental unit of life, which will be helpful to you for understanding this chapter better. Okay, now today further we are moving with another practical, or we can say another experiment of plasmolysis. What do you mean by plasmolysis? I already explained in my previous video what is plasmolysis. Then also I would like to tell you the definition of plasmolysis is it is a process. Plasmolysis is a process of shrinkage or contraction of the protoplasm of the cell. What do you mean by protoplasm? I told you that protoplasm is the cytoplasm plus nucleus. Okay, so it is the contraction or shrinkage of the protoplasm of the cell. Why this contraction occurs? Due to that the cell loses water. As cell loses water, the protoplasm of the cell contract, or we can say shrink. This phenomenon or this process is called as plasmolysis. So we have to learn this plasmolysis in this, we are showing the plasmolysis in a plant cell, that is an onion cell. Okay, so we are doing the activity of plasmolysis of an onion cell. For understanding plasmolysis, we are using the rheolith. The image you can see over here, dear students. This is an image of rheolith. Why rheolith is required for understanding plasmolysis better? Because you can see over here that rheolith is colored. Okay, it means that whatever the cell sap is there, whatever the cell's cytoplasm is there, it is colored. So you can easily observe plasmolysis. You can easily observe the shrinkage of the cell sap under the microscope. But as I do not found such kind of leaf or such kind of leaves nearby my area, so I am giving you and I am showing you plasmolysis in our onion cells. Getting, dear students? So for doing this activity, the materials required, that first of all we are using onion. Okay, we previously also did the onion cell in how to observe plant cell and how to observe onion cell under the microscope. So here for plasmolysis also we are using onion cell, because onion cells are easily seen under the microscope also. And the second thing is that we can easily peel out the onion epidermal layer. So for that we are using the onion. Secondly, we are using the blade or cutter to remove out the onion peel. Then the next is we are using the dropper for taking out the iodine solution. Now why we are using iodine solution? Iodine solution is a stain. As I also previously told you that we are using such stains to color the inner part of the plant cell. Now as the inner part of this cell is colored, we can easily show the plasmolysis, we can easily show the shrinkage, we can easily show the contraction of the cytoplasm, or we can say of the protoplasm under the microscope. So for that purpose we are using such stain. We are also using the distilled water to wash or to keep wet our epidermal layer. Then the main thing over here, we are using the solution, salt solution. You can use sugar solution also, you can use salt solution also. Now why the sugar solution and salt solution are can be used over here, dear students? Because we have to shrink the cell, means we have to lose the water of the cell. So for that purpose we are using here salt solution. Salt solution means what? Salt plus little amount of water. Okay, now what happened when we are keeping our epidermal layer of onion cell into this salt solution? Due to the salt solution, concentrated solution is there outside of the cell, and inside there is less amount of salt is present, or we can say less amount of NaCl is present. So what happened? Cell loses the water, and as a cell loses the water, cell get shrink, and then we have to observe that cell under the microscope. Secondly, we require a clean glass slide, and we require a cover slip, and our most important thing, we require a microscope. Okay, so let's begin with it. this practical, dear students. So first of all, what we have to do, we have to remove out one inner layer of the onion peel, and with the help of blade or cutter, we have to take out the epidermal layer. We have to gently remove the inner part of the onion peel. Okay, you can observe, see over here, dear students, this is a very translucent, thin epidermal layer of the onion cell which we had removed out. Now I am keeping this peel, epidermal layer of onion cell, into the salt solution for 10 minutes. For how many minutes? 10 minutes. Now, as I already told you why this step is very necessary, as the salt solution is there, due to the concentration salt solution is outside of the cell loses some amount of water from it. So we have to keep this, our epidermal layer, into salt solution for 10 minutes. Okay, so let's come after 10 minutes, dear students. Almost 10 minutes are completed that we had kept our pill into the salt solution. Now it is time for preparing the slide. So for preparing slide, first of all, we have to keep 1 or 2 drops of water. As I told you, why we have to keep a drop of water, to keep our onion peel in wet position. Okay, after putting a drop of water, we have to take 1 or 2 drops of iodine solution on our clean glass slide. Okay, I have taken 2 drops of iodine solution. Okay. After adding drops of iodine solution, we have to remove out the onion peel from the salt solution, and we have to keep it on our glass slide. We can use paint brush also here, dear students. Okay. Now extra amount of iodine solution and water I am absorbing by this paper. Okay. Now put the cover slip. Make sure that bubbles will not come. Yes. Now our slide is ready to observe under the microscope. So now we are observing the slide under the microscope. Now you also get clear idea, dear students, that how to observe a slide under the microscope. First of all, we have to take the microscope. We are with this illuminator. You can see the mirror at the below portion of the microscope. With the help of illuminator, first of all, we have to adjust the light. We are observing this onion peel at the low power. At the low power means at 10x. You can see this is the lens on which it is written as 10x. So it is the low power observation, or we can say low power microscope. So we are observing this onion peel under the low power. Okay. After keeping the slide on the stage of the microscope, we have to keep these clips to fix our slide. And now with this coarse focusing, we are observing our onion peel, and we will observe plasmolysis under the onion cell. This is the look under the microscope, or we can say this is the image we can observe under the microscope. Okay, if you remember, dear students, when we were observing onion cells previously, and that if you remember that all cells are combinedly together with each other, nucleus and cytoplasm we can observe. Okay, now you can observe over here, dear students, that each and every cell has little amount of space between cell wall and cytoplasm. You can clearly see over here in this image, dear students. So what is this space? Okay, why this space is coming into this particular image? So you can observe, dear students, that as we had kept our epidermal layer into the salt solution, due to that salt solution, plant cell, or we can say onion cell, loses some amount of water in the surrounding medium, and as plant cell loses some amount of water, shrinkage of cytoplasm will occur, and you can easily observe over here the shrinkage of cytoplasm, that all cell having their cytoplasm is somewhat little bit far away from their cell wall. You can easily observe cell wall over here, and you can easily observe the shrinkage or contraction of the cytoplasm over here. Or you can also, dear, observe over here that all the nucleus of the cell are not centrally located, they are little bit somewhat at the side. So this is the activity, how to observe plasmolysis. What is plasmolysis? Now the idea is clear to you, dear students.